So welcome back again to our continuing look at the diversity of living organisms as we find them on our planet. Today we're looking at what has been the most problematic chapter for me this time through, because the fungi have been undergoing a major reorganization. This has been brewing for about the last 10 years or so. There's been some rethinking about both the origin of fungi and the taxonomy of the kingdom fungi. And that has sort of shaken itself out over the last few years and is now reflected in the textbook. And so what you are finding and what you will find is that there's a discontinuity between what you saw in the lab manual, which is based on the old taxonomy of kingdom fungi, and the new taxonomy as described in the textbook. We will focus mostly on the new taxonomy identified in the textbook in this presentation, because that seems to be the direction in which mycologists, the people who study fungi, seem to be headed in. And so one of the reasons why this chapter has been problematic is because, number one, I've had to rewrite this presentation, but also number two, I've had to rethink and research all of what I thought I knew about fungi to make sure that I had squared away everything that I wanted to present in the presentation today. So bear with me if I seem a little bit unsure at times. It's simply because this is the first time in some cases that I'm presenting some of the information that's here. And so it is a little bit new to me as it is new to you, but we'll make the best of it as we can. I think I'm pretty much satisfied with what I've got written up in the PowerPoint at this point. And I feel reasonably confident that I can discuss the new taxonomy with some degree of confidence, but we'll see how we get on. So one of the real issues with fungi is that over the course of perhaps 150 to 200 years, just exactly what constitutes a fungus has changed. The traditional definition of a fungus always included the statement that there was no flagellated stage. And that was one of the reasons why things like the chytrids, a clade that we will encounter this morning, were specifically excluded from the kingdom fungi, because they all have a flagellated stage. But we now know from DNA evidence that they really are true fungi. They are basal to much of the rest of the kingdom, but they are still most definitely true fungi, even though they have a flagellated stage. We now also recognize a couple of clades that are even more basal than the chytrids that also have flagellated stages. And in some cases, well, actually in both cases, these two clades are still not confirmed as true fungi, but they are at least suspected to be very closely related to fungi, if not actually part of the clade. So they are placed basal to the clade, and as you'll see in the phylogenetic tree, they are connected into the base of the tree by dotted lines, indicating that this is where we think they belong, but we have yet to fully confirm that this is really where they do belong. By the same token, you may recall when we talked about protists, that there were a group of protists called the oomycetes. These were the water molds and downy mildews. And the reason why they have the designation mycete is because at one point they were actually classified as fungi. And yes, they do have, in the case of some of them, a flagellated stage, but it was considered that because of the structure of the body of the mature form, made up of thin threads known as hyphae, which is very characteristic of all fungi, that these were actually probably fungi and not protists. That has changed. We now classify them as protists. They still retain the mycete label oomycete, but they do not belong in kingdom fungi anymore, at least not under the current phylogeny. So there's been a lot of rethinking what constitutes a fungus, and that has in turn led to clades moving in and out on the basis of the thinking. What you're also going to see in the phylogenetic tree, and I'll discuss this a little bit without getting into too much detail, is that one of the previous major clades has now been split into two, and some fungi that were once part of that major clade, then moved out, have now been moved back in again to constitute part of one of the two new clades that have been derived from the previous clade that's now been split into two. Is this getting confusing? Yeah. And if you're feeling a little bit confused right now, don't panic, because it is what the state of most phylogeny has been over the course of the last 30 or 40 years, as people have come to terms with the phylocode and cladistical analysis and using DNA sequences as the primary way of establishing relatedness. When we get to the animal kingdom, which is the last kingdom that we have to cover, it's the one that's left after we finish fungi, we'll even see that there's been some major rearrangements, not so much in the phylogenetic tree as it's presented in the textbook at the moment, but in the past, and we'll touch a little bit on that as well. So yeah, one of the real hassles with teaching the general biology 2 class, and specifically the phylogenetics and taxonomy, is because this is probably the most dynamic part of biology, because of the ability to access so much DNA information now with the increased ability to sequence genes. And does that mean there's going to be more changes? Almost certainly. Probably not much that you have to worry about, but something that future generations of students and future generations of instructors are probably going to have to struggle with as well. Anyway, enough of that. I digress. Let's move on and start talking about fungi and what makes them so unique and so interesting, because they truly are very interesting organisms. So one of the most amazing things that has been discovered in about the last 20 years is that the largest organism on the planet, by any measure, whether you're talking about volume, whether you're talking about land area occupied, whether you're talking about mass, to some degree, even if you're talking about age, happens to be a fungus. It's called Amylaria ostoyae, otherwise known as the honey mushroom. I think I prefer that underlies a huge aspen grove in one of the national forest regions in Oregon. There are several of these very large fungal growths scattered through the Pacific Northwest. This happens to be the largest of them. And it covers a huge mass of ground, except that if you were to walk through the aspen grove that it underlies, you would probably not even realize it was there, because the vast majority of the fungus is subterranean, it's underground. And the only evidence that you really see of the presence of the fungus is that periodically mushrooms appear, because it belongs to that clade of fungi that produce mushrooms. We'll talk about those later in the presentation. And so periodically you see mushrooms appearing, which is the first clue that there's something underground. The reason why we know this is a single fungus, and this is quite amazing, is because people have taken samples of the fungal mass and done genetic screening on it and recognized that they all share the common genetics, no matter where the samples are taken from. And also by radioisotope studies, introducing radioactive isotopes into one region of the fungus, and then tracking the movement of that radioisotope throughout the full body of the fungus. Because one of the things that we know about fungi is that the mass of the fungus forms one continuous body, and things are moved throughout that body by a process called cytoplasmic streaming, which means that anything that's taken in through one part of the fungus as a food resource is shared with the whole of the fungus through cytoplasmic streaming. And that also includes its radioactive isotopes. And so we have demonstrated pretty conclusively that this is a single large fungal mass. And as the note here says, it weighs many hundreds of tons and it's old. The note here says 1900 years. I've actually come across articles on the internet that suggest that it may be even older than that. It could be close to 2,500 years old. So I guess the age of the fungus varies because it's kind of hard to predict or determine just how old a fungus is. We can figure out how big it is, but actually knowing its age is a little bit harder to determine because the only way you can figure that out is to extrapolate backwards to when you think it might have represented a single point of origin. And that is somewhat difficult to determine. But this just gives you a clue as to just how extensively fungi can penetrate into their environment. And the reason why is because of the makeup of the fungal body. We'll get into that in a few moments, but let me give you a little bit more information as background to the fungi. So there are a substantial number of fungal species that have been described, but there are people, mycologists, who make a living going out trying to find new fungal species. And basically, no matter where they go and no matter how carefully they look, they always encounter new fungal species. Basically, fungi have a distribution that rivals that of protists. Doesn't rival that of prokaryotes. They have the widest diversity of any group of organisms on the planet. But fungi as a group seem to have a distribution almost equivalent to that of protists. And to some degree, in terms of diversity of species, they may border on a diversity that is along the lines of insects. We are really puzzling about just how diverse insects are. I've seen estimates of anywhere between 5 million and 10 million species of insects. Fungi are getting up there in terms of estimates of species. And one of the things that we also need to bear in mind is that they come in many forms. Now, you are familiar with at least one type of unicellular fungus, yeasts, and our ability to use yeast for our own benefit. We use it for baking bread. We use it for making adult beverages, alcoholic beverages, because they carry out fermentation, and they do fermentation in both instances, and they are unicellular fungi. And we'll encounter several clades of fungi that are basically unicellular, and they are basal to the rest of the kingdom of the phylum. Kingdom. Let's get it right. But the vast majority of fungi have at least large bodies made up of very thin threads called hyphae. They reproduce through spores. So if you recall the plant life cycles that we were talking about in the previous two chapters, remember that spores are the product of meiosis in plant life cycles. The same is going to be the situation here. What makes fungi somewhat unique is that the vast majority of them have both a sexual reproductive cycle and an asexual reproductive cycle, both of which produce spores. And so it is the spores that enter into the environment, and those spores are uniquely, or universally, I should say, haploid. And when they germinate and give rise to the new fungal body, that new fungal body is haploid itself. Now, you're going to find in the life cycle, as we discuss it, that there is going to be something very unique about the haploid nature of the fungal body. And we'll talk more about that when we get to the life cycle. But here again, we're encountering organisms that break our knowledge of life cycles if we are thinking in terms of animal life cycles. Again, we've got to remember that the animal life cycle, as much as it's the life cycle that we want to focus on, is not the norm. It is, in fact, the unusual life cycle, and we need to take into account that other organisms that are not animals do not share an animal-like life cycle. In fact, it's anything but an animal-like life cycle. So let's talk about some of the characteristics that bind fungi together. So these are characteristics that are uniformly found across all fungi, irrespective of whether they are unicellular or made up of these large hyphal masses. And you'll notice that I'm trying to avoid using the term multicellular, and you'll understand why when we talk in a little bit more detail about the nature of the fungal life cycle. Because to say multicellular does not necessarily always apply, even if a fungus is made up of these hyphal masses. So one of the things that we know about fungi is that they are heterotrophs. They do have to get their carbon source and their energy source from organic molecules. But unlike you and I and other animals, they don't ingest their food and then digest it internally. They secrete enzymes into their environment that begins the process of breaking down the molecules of the food resource that they're tapping into. Then they absorb the resulting products into the body of the fungus. So that means for a fungus to be successful, the body has to be adapted and to specialize in the process of absorption. And what you're going to see as we look at the nature of the fungal body is that it is highly adapted to the process of absorption. And this is, again, what makes fungi somewhat unique. They also have diverse interactions with their environment and with other species around them. So we primarily recognize them as decomposers. And that makes them really important because they recycle organic matter back into the environment to make it available for plants and other organisms. But one thing that really makes them unique, or mostly unique, is that they are one of two major groups of organisms that have the capacity to digest cellulose. Now, if you've forgotten about cellulose from Biology 1, let me remind you that cellulose is the primary material that makes up plant cell walls. And as far as animals are concerned, and the vast majority of life forms, it is indigestible. So when we talk about soluble fiber or fiber in general as being a component of our diet, what we're really referring to is the cellulose fibers that we get from fruits and vegetables in our diet. We can't break that down. It's indigestible. So it passes through our gut and it contributes to the volume of our fecal matter. And it allows it to retain moisture so that it moves smoothly through our digestive tract. It also influences what sort of molecules we can absorb from our food and what sort of molecules we don't absorb from our food. We know that it has an influence over our general health and well-being. But if you want to break down cellulose, then you either have to be a prokaryote, typically an archaean, or some species of bacteria, or you need to be a fungus, or you need to have some sort of relationship with an archaean or a fungus in your gut in order to be able to decompose cellulose. So when you think in terms of diverse organisms as termites and cows, they have fungi in their guts that help them process the cellulose in the plant material that they're using as a food resource so that they can get nutritional benefit from eating, in the case of cows, grass, in the case of termites, the wood framing of your house. Then we also know that fungi can be parasites. They can be parasites of animals. They can also be predominantly parasites of plants. We know that a substantial number of agricultural losses are attributable specifically to fungi. And if you've ever been to the supermarket and picked up a punnet of strawberries, a box of strawberries, and looked underneath and seen white fibrous material growing inside the punnet, well, that would be a fungus called Rhizopus, which hopefully you encountered in Bio 1. You may have had an opportunity to look at some slides, the reproductive structures of Rhizopus. But here is an example of a fungus that is acting as an agricultural parasite. And it is estimated that in some circumstances, upwards of 80% of agricultural losses are due strictly to fungi. So they are, for humans, important parasites at the agricultural sense, but also they serve as parasites in other ways. And we know that there are a number of human diseases that are caused by parasitic fungi. And then, as we'll also see, there are some extremely important mutualistic relationships between fungi and other organisms, predominantly algae and cyanobacteria in the form of lichens, but there are also the very important mutualistic relationships between fungi and the vast majority of the plant kingdom. We suspect that somewhere around 90 to 95% of all members of the plant kingdom as we recognize it right now have some form of mutualistic relationship with fungi. And those relationships are important and they're extremely ancient. You may recall that I mentioned that we would discuss this a little bit in this chapter when we were talking about the origin of the plant kingdom. And we'll address that a little bit later as we move through the presentation. So what is the big deal about the body structure of a fungus? Well, the vast majority of fungi are made up of these filaments known as hyphae. And it's the filaments, the hyphae, that make up the body of the fungus known as the mycelium. So when we look at a typical fungus that you might be familiar with, say one that produces mushrooms, underground, you're going to see all of these white thread-like structures. These are the individual hyphae. And they are very, very fine in diameter. They're actually finer than a human hair. And the fungus can produce thousands upon thousands upon thousands of these hyphae to penetrate into volumes of soil. This is the reason why fungi are so good at doing what they do, because these very, very fine threads can penetrate in large masses into the food resources that they're utilizing. So I've included in the note a little bit of information about that. And so it points out here that in 1 cubic centimeter of really good soil that contains a lot of organic matter for the fungus to utilize, there could be upwards of 1 kilometer of hyphae. Let that sink in for a minute. A cubic centimeter, so 1 centimeter by 1 centimeter by 1 centimeter. The total mass of fungi can constitute a length of 1 kilometer, 1,000 meters. So that's just over half a mile, if you're unfamiliar with the idea of a kilometer. And the surface area attributed to all of that hyphal mass is more than 300 square centimeters. Now, I can't give you a non-metric equivalent, but that's a large, there are 100 centimeters to a meter, so that's 3 meters by 3 meters. So rough conversion, that's about 11 feet by 11 feet. So that's a pretty large surface area, and that is because the fungi structure is adapted to absorption. and absorption works with large surface areas. And so that's where we start focusing on the hyphae. The hyphae actually create a very significant surface area to volume ratio, a large surface area compared to the volume. And the reason why that is the case is because volume increases by the cube of the radius, while surface area increases by the square of the radius. So if you increase the volume of something, the surface area does increase, but the volume increases faster than the surface area. So if you keep the volume very small, then the ratio of the surface area compared to that volume is quite large. And so one of the things that we notice about fungal hyphae is that when they grow in length only. They don't grow in girth. So they don't get any thicker as they grow in length, and they can grow very, very quickly. And because they're so fine, you can pack hundreds, thousands of them into a very small volume, which only means that you are packing in a large surface area for absorption. And again, this is an adaptation of the fungal body for the process of absorption, because that's what they specialize in terms of obtaining nutrition. And the other feature that we need to remember that is associated with all fungi is the presence of chitin as the cell wall material. And chitin, hopefully you remember, is the same material that makes up the exoskeleton of insects and arthropods in general. And so that material is actually edible. So when you consume fungi in the form of mushrooms, morels, or other types of fungi that you might choose to consume, because we do eat a substantial range of fungi, the cell wall is very digestible. We can break down chitin. You may have learnt in Bio 1 that in fact we take advantage of that by turning synthetic chitin into a surgical thread that is used for sewing up tissue subcutaneously, so that you don't have to go back in and take out the surgical thread. Your body digests it away very slowly as the wound heals. And so that just reinforces the fact that chitin is very digestible, which means that fungi can serve as a very useful nutritional source, not only for us, but for animals in general, because animals can break down the chitin. So I'll go back to this slide for a minute. If these numbers don't impress you, when I was teaching my plant physiology class over at UCF many years ago, I did a little bit of research on fungal masses, because it's important in plant physiology to talk about the interaction between plants and fungi. And I came across a paper that was published back in the 1940s, where a research group decided to check and see just how much fungal mass could be produced in association with a single prairie grass. So they inoculated the seed of a single prairie grass with some fungal spores for the fungus that was known to associate with it, and planted it in a wooden box that they had built themselves that was 1 meter on edge. In other words, it was 1 cubic meter in volume. So 1 meter by 1 meter by 1 meter. And they allowed the prairie grass to grow in there. And obviously the fungus germinated and developed into the fungal mass in the soil. At the end of the prairie grass's growing season, they then determined how much fungal mass had been produced. I'm not sure exactly how they did this, because they didn't detail in great detail exactly how they measured everything. I think a lot of it may have been estimated, but they tried to determine in terms of length how much hyphal mass had been produced, and then in surface area how much hyphal mass had been produced. So in the 3-month growing season that the prairie grass was alive, because that's approximately how long it lived, in this 1 cubic meter of soil, the fungus produced over 500 kilometers of hyphae with a surface area that was measured in tens of square kilometers in a single cubic meter of soil in a 3-month period as the fungus grew in conjunction with its symbiotic host, the prairie grass. It's quite an amazing volume of material, but that just reflects that the fungus is geared towards absorption, and it does so exceptionally well because of the nature of the fungal body. We'll return to this idea a little bit later in the presentation. When we start looking at the hyphae, there are 2 basic forms for the hyphae. So some hyphae are actually divided up into individual quote unquote cells, but some hyphae lack that structure, and we refer to those as cenocytic or aseptate. So the reason why I say quote unquote cells is because the cell walls always have a hole with them to allow direct cytoplasmic connection between the adjacent chambers. This is to allow cytoplasmic streaming. So a septate hyphae does have a separating structure called the septum. It's made out of chitin, the cell wall material, but it has a hole through the center of it to allow direct cytoplasmic interaction. Other hyphae lack those connections. These are the cenocytic or aseptate hyphae. And so this is a large continuous cytoplasmic mass that is multinucleated. And these 2 types of hyphae, when we start talking about reproductive processes and life cycles, will become important, because the presence or absence of the septate do determine the way we describe the hyphae. And we'll actually address that in a moment or 2, but we'll talk more about it when we get to the life cycles. Then we also know that some fungi carry out specialized processes in terms of just how they go about obtaining their nutrition. So quite remarkably, some fungi act as predators. They have specialized hyphae that allow them to trap small nematode worms moving through their environment. And here you can see a nematode worm that's been captured by the hyphae of the fungus. It will now secrete digestive enzymes into the environment that will digest away the nematode over time, and the resulting products will be absorbed into the body of the fungus and distributed by cytoplasmic streaming. But then there are a small group of fungi. They have been referred to as glomeromycota or glomeromycetes, and we will talk about them a little bit later in more detail when I identify the clades, that form a more intimate relationship with fungi and specifically with plants, and specifically with plant roots, by creating an extension of the hyphae that lies against the root, referred to as a haustorium. And this forms what is known as an arbuscula. Let me turn on the pen. Arbuscula. No, it's I remember my spelling. Arbuscula mycorrhizae. Mycorrhizae refers to the interaction between fungal hyphae and plant roots. In this case, the arbuscular term refers to the fact that you've got these haustoria penetrating in through the plant cell wall. But what is important to bear in mind here is that it does not penetrate into the plant cell proper. So it doesn't penetrate through the plasma membrane of what is referred to as the protoplast. That's the plant cell that lacks a cell wall, or the part of the plant cell that excludes the cell wall. It's referred to as a protoplast. And so the fungal extension, this haustorium, secretes cellulase, an enzyme that breaks down cellulose, that allows it to digest its way through the cell wall, and then it creates this very highly branched haustorium that lies up against the plant cell membrane. And this allows for a large amount of transfer of material backwards and forwards between the fungus and its mutualistic host, because this is a mutualistic relationship. The fungi transfer water and nutrients, particularly minerals, from the soil that it can import much more easily than a plant root can. And so it provides those to the plant. In return, the plant provides carbon sources, products of photosynthesis that it transfers to the fungus. Now, these interconnections between fungi and plants, whether they be of this arbuscular type or what is referred to as an ectomycorrhizal structure, which is external to the root and just forms a mesh around the root, these connections can also connect plants together. And these connections between the plants mediated by the fungi have become known as a word, sorry, let me back that up, wood wide web. And there is constant communication going backwards and forwards between the plants mediated by the fungi. And we know that when for instance, a plant gets attacked by a herbivore, it signals that attack, and that signal can be transmitted via the fungi to surrounding plants, and they can prep themselves and protect themselves from potential herbivory, insect attacks and things like that in response to the signals received by received through this wood wide web. Likewise, adult mature trees can provide support to younger trees that are unable to support themselves because they are in the shadow of older trees or are in some way disadvantaged by being adjacent to older trees. The older trees can actually distribute materials through to the younger trees via the fungus. As the younger trees mature, they can then return the favor by sending resources back to the older trees. And so there is a certain degree of reciprocity in this relationship being mediated by the fungi. I wish we had more time to talk about this, but we don't. But I will put in the resources section of the Canvas page some links that you might want to chase up on and a reference to a book that will allow you to learn a whole lot more about fungi and their interactions with the plant kingdom if this is piqued your interest in fungi. So take a look at the resources section if you want to find some more information regarding this, the whole nature of this interaction between fungi and particularly plants, but that in the case of the book, it goes into a whole lot more detail about all sorts of different things about fungi. So this is just again referencing the two different types of relationships. So you've got the ectomycorrhizal, the sheath around the roots, and then the arbuscular mycorrhizal structure that was shown in the diagram previously. And we'll look at another picture of an arbuscular mycorrhizal structure a little bit later in the presentation. So let's take our first break. We've been going for a little while, and I will stop the recording at this point after giving the concept check question, and I'll see you on the other side of the break with the answer to the concept check question. So here's the question. Compare and contrast your nutritional mode as an animal, obviously, with the nutritional mode of a typical fungus. I'll see you on the other side of the break.